sorry for the delay. There were a couple of things that we had to sort out, but I think now we are all set and ready to start. I'll be very briefly, the idea is just to give you some introduction and make some announcements. First of all, breakfast, coffee breaks are going to be offered here. So just go outside, you cross here this little bridge, and you have the poster area, and the coffee is going to be in that area. If you haven't displayed your poster and you're going to make a presentation, please use also the coffee break to go there and hold your poster. We have a couple of situations that might need some more specific orientation. For instance, foreigners, you have received a voucher, so for meals. And there are, I think, 3 places that you can use. One is the cantina of the campus. The other is another restaurant in the area. And it's specific because you have your names on it. So you probably cannot exchange to people unless you go together. I don't know all the details. If you have any trouble, please contact Humberto or Thiago because they are going to be very good at helping these things as well. So just remember that there are some restrictions. It's a new regulation, if I understand correctly, from the founding agency, and they have changed it a little bit. So Brazilians have a different policy than foreigners in the sense because you really have to use the vouchers. I'm sorry if there's going to be some troubles. I was not aware of it until we arrived, so we are trying to sort out, and this is the piece of information that I got to transmit to you. If I'm not wrong, this is, let's say, the key message that we are supposed to provide you. If there are any other details, just please ask me or ask anybody from the Institute. Remember that I'm also a guest here. Both myself and Rolf, we are not from this Institute, so we also have to try maybe to get the information for you. But it's not a problem. We are here very happy to welcome you and to help you whatever you need. I hope you can enjoy this week. So I'll briefly just go to a couple of slides to remind you the beginning of these schools. Actually, we started 10 years ago. This was the first school. And in this time, it was organized by myself, Rudy Podgornik, and Roland Letts. At that time, Rolf was an invited speaker, but he gradually started to be involved in the organization, and actually he was the person that was helping quite a lot. So in the next school, he changed it from being just a lecturer to be one of the main organizers, and that's the beginning. So this was the first school. I should also mention that, unfortunately, we lost Rudy Podgornik last December due to COVID. It was a very sad moment to all us. And it's important to bring his name back because he was actually the first person that incentivate, not just incentivate, but catalyze this school to happen. I met him in an event in Trieste. It was Physics of Virus, the name of the event. We were discussing things. This was back in 2012. And in that meeting, he said, well, we should organize a similar meeting focused on biological physics in Brazil. And we started talking with different people, and then at some point, we ended up with this event. So it's just a pity that he cannot join us any longer, but I'm sure that he was going to be very happy that we continue his job or his idea, and we can go further together, especially trying to provide to the new generation a view of what we can do in this field. The second event happened in 2017. Now we had Rolf as one of the lecturers and our organizer as well. There was another edition in 2020, and this was the last event that we had together before the pandemic started. So this was the reason why we have these long breaks. And now we are back. So there were a couple of things that changed. You probably have seen that Cecilia cannot join us. And also there's going to be another change because Rosangela that appears here, unfortunately, she also had some issues and she cannot be present. But she asked 2 other colleagues that work in the same field to be present, and they're going to deliver the experimental part. So this was the program that we have available on the homepage. I think there is an updated version right now with this update that I just mentioned to you. So just remember that here, instead of Rosangela, we have Achilio that's present somewhere, and Luis Fernando that's also present somewhere, I guess. So there are going to be 2 talks from Achilio and 1 talk from Luis Fernando. So the program is going to remain more or less the same. You are probably wondering what this discussion study time means. This is a free time, essentially, that you can use in different ways. You don't have, let's say, official classes, but it's a time that you can talk to one of us to discuss specific questions, to bring your projects for specific discussions. Maybe you need a support from machine learning. You can contact the lecturers that are present. Maybe you were wondering about more experimental details or more details about the class. So try to use this time as better as you can. It's a moment for us to really interact and like a more spontaneous thing. We used to do in a more organized way in the previous schools, but it seems that now that we're a bigger number of people, it's going to be better that we make a little bit more flexible. So you're free to contact any of us and say, yeah, I would like to discuss with you. Just try to be attention that part of the people, part of the lectures are not going to be available the whole week. So give more, let's say, preference to people that are giving the class in the same day, because this helps you to really make sure that you can contact this person, okay? I guess this is all for the instructions. There is just a minor detail that can be relevant if you need the certificates. That's going to be attendance sheet, and you have at least to fulfill 80% of your frequency. So if you're going to hang out during the evenings, remember that the class, they are back 10 o'clock in the next day. What else? It's also nice to see where we're coming from, so you can see that this school is quite diverse. Although the number of participants that are not Brazilians have decreased at this time. I don't know the reason, but it has decreased. But we have people from different nationalities and different languages. So try to use this as another opportunity for you to get to know each other and to do a very nice network, because we can all learn from the participants as well. So this is also a relevant aspect of this school. And remember that this is a school. So being a school, it means that everybody's here to learn. So try to be very respectable, respect people, try to really remember that even the presentations of the students sometimes, they are not in the professional level. We are here to help everybody to grow together. This is the key message. So remember to keep this in mind while you're interacting with everybody. I guess this is all. We also would like to thank people that without them, you know, this school would not exist, both people that support the financial part, and I think the even more important is the people, the humans that are really helping us to organize and to host us here. I just named a couple of them here. There are many more. You can see just outside, and I think they really deserve our support. Our thanks for their support. Okay? I guess that's all from my side, at least. It's just really to give this brief message and welcome you. I'm going to give the stage to Professor Chuck Farah. He's going to give you the introduction of, let's say, the first real class. And we have a format that's really very a class-like format. So you're not going to have like a chairman that's going to be giving questions. So feel like you are in a classroom and totally free to interact with people. If you have any question, just raise your hand, either to me or during the class to Chuck. Okay? So, Chuck, please, the stage is yours. We have a problem to change this. So, this one.
Now it's working. Yeah. Huh. Okay. Good morning, everybody. It's very nice to be here. Fernando mentioned to me that it's a mixed audience, so that's why I'm giving a lecture, or my lecture is going to be sort of on basic protein structure. Because it seems that maybe some of you are like trained biochemists or biologists, but others maybe just physicists and have never taken a biochemistry course. So maybe I'd like to ask you, Who here is trained as physicists? Okay, that's about a third maybe. And what about how many of you are biologists? That's another third, maybe. And so like others are like chemists or oh, there's chemists. Okay, so depending on your course, you probably had some kind of biochemistry course. So this might, but maybe not too many of them. So this might be a good revision of some basic concepts. So I'm going to talk about basically these four topics here about the structure of proteins, basically, and the concepts can be extended to the structure of nucleic acids and carbohydrates and lipids and other biological macromolecules. I'm not going to talk about protein folding or protein dynamics because I think you're probably going to have a lot of lectures specifically about this during the whole course. But the first thing I'd like to do is to show a video, and let's see if this video is going to work. It's on YouTube, basically. It's a YouTube video, and it just basically shows how what are enzymes, okay? And then I'm going to go back to some basic concepts, but I think it might be just sort of interesting to have an idea of the importance of proteins and enzymes in biology. So let's see if I can get this thing to. And I'm not sure if the sound's going to work. If it doesn't, I'll try and narrate. And yeah, so this video is on the PDB site. It's on the protein database site. They have sort of like educational videos. Yeah, sure. So this is just, I think this video is called How Enzymes Work, or something like that. Yeah. Okay, so can we make it like the maximize the.
Every single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes.
Notice how crowded it is in the cell.
An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur. To understand how enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitate. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron-sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell in every living creature on the planet.
Okay, so what was important in this video that I showed you was just to show you some sort of obvious general aspects of that video. You could see that the cell environment, the molecular environment, is very crowded. Okay, there's lots of, and there's all of the molecules are moving all the time. Even the enzyme itself, the catalyst itself, is dynamic, so it's fluctuating in its structure. So, and all of these reactions are happening under very, very mild conditions. Okay, so some of you have worked in chemistry laboratories and you've learned to make a chemical reaction happen. Sometimes you have to raise the temperature to very, very high temperatures, or you have to add solvents. The reaction only works in chloroform or in carbon tetrachloric, some kind of organic solvent or something like that. But all of these reactions in our cells, they occur in an aqueous environment, more or less, at a normal temperature. Almost all the reactions occur at the same temperature, in fact. So Mother Nature, over millions or billions of years of evolution, has been able to design macromolecules, mostly proteins, to catalyze selected reactions in a very, very regulated manner. And that's the great challenge for us in biomedicine in order to understand this relationship between the structure of proteins and the reactions that they catalyze so that we can manipulate them. We can either try and inhibit them or we can try and design new proteins with new functions. So that's the great challenge, okay? And that's sort of shown here in this slide, where we know the central dogma of molecular biology that was proposed by Francis Crick in the 1960s, that DNA codes for RNA, that codes for proteins, okay? So the sequence of the DNA is transcribed into RNA molecules or messenger RNAs. These messenger RNAs are translated into proteins using the genetic code. But then the next step is trying, from the sequence of a protein, how does it actually fold up into its three-dimensional structure? And then how does that three-dimensional structure determine what its function is in the cell? And these are still big challenges for us, okay? There have been great advances in this part here that probably maybe Fernando's going to talk about. I might touch upon it later on. How do you predict the structure of a protein from its sequence? But then how does that structure translate into function is still very, very difficult to understand unless you can actually watch the protein executing its function. So, but what I'm going to try and do in this first lecture is to just talk about the basic concepts of protein structure. Okay, so let me see if this thing works here. Okay, so as the video showed, cells have a crowded environments inside of them. There's lots of macromolecules. Some of them are globular in shape, that is sort of like spherical, with three dimensions that are more or less in the same order of magnitude. But then there are some other molecules like DNA or fibrous proteins or carbohydrates that are more extended. They make fibers, sometimes executing structural functions, like maintaining certain structures in the cell intact, and sometimes sort of providing trails in which other proteins can sort of like highways in the cell, where other proteins can walk along these fibers to get from one cell to another. Okay, and there's a lot of water in the cells, and you can see that the macromolecules are surrounded by water. So the interactions between the macromolecule and water are very, very important to understand the structure of the macromolecules and how they interact with other molecules in the cell. Okay, and when we represent the structure of proteins or DNA, as shown here, we sometimes use conventions to show different elements in the molecules. So, for example, it's very, very common that nitrogen atoms are shown in blue, oxygen atoms are shown in red, and usually the nitrogen atoms, they're often, not in this case, but in proteins, they're often associated with protonated amine groups, and they're often positively charged. So sometimes when you see a blue surface on a protein, it might sort of indicate that it might be a positively charged area. And oxygen atoms are very electronegative, and they often are associated with acidic groups, and they're often sort of negatively charged. So you sometimes see protein surfaces colored with this, and the red regions are often the more negatively charged regions, and the blue is the more positively charged regions. Here, the yellow are phosphorus atoms because these are nucleic acid structures, and the carbon here is shown in white. But the carbon can be many other colors, often green or black. Okay, so you can represent macromolecules in many different ways. Giving like the volumes of each atom can be shown as a volume. Sometimes they can be shown as stick models, sticks like lines between the atoms, etc. Okay, but let's go down back to basics. What are proteins? Since proteins are the main catalytic entities in cells, they're often the major focus of biomedical research, trying to understand how proteins work. So what are the building blocks of proteins? Building blocks of proteins are amino acids, and there are 20 naturally occurring amino acids that are coded by the genetic code, and they're selected to be incorporated into proteins in a process called translation that's catalyzed in ribosomes. Ribosomes are these large, I think I showed them here. This is a ribosome here. This is a small subunit of a ribosome, and it binds to the messenger RNA, and then a large subunit, which is like maybe twice as big, it's around here, That's where the amino acids are brought to it by transfer RNA molecules and condensed together to make a polypeptide chain. Okay, and these are the amino acids. So there's 20 of them, and you can see that they can be let's say they can be categorized according to in different ways. Okay, here they've been sort of categorized based on their chemical properties. Okay, so we have aliphatic amino acids in the top row here that have side chains. So all of them here, they all have a nitrogen atom. Okay, what they call an alpha carbon. Okay, and a carbonyl group, which is a carbon and an oxygen with a double bond here. Okay, so this is a carbonyl group. The hydrogen atoms are not shown here. Okay, so the nitrogen, we'll see this later. The nitrogen has a hydrogen bound. Okay, and this alpha carbon here has a hydrogen bound. Okay, so but we're basically we're focusing on the quote unquote heavy atoms in the protein. Heavy atoms in proteins are not like they're not heavy atoms as in metals, transition metals or anything like that. They're just any atom that's not a hydrogen is considered a heavy atom for protein in protein structure language. Okay, so here we have these aliphatic. So these are the side chains. These amino acids differ by the nature of the side chain. So you can see we have some amino acids that have side chains that are just made up of carbon groups. Okay, so this would be a methyl group, and this would be an isopropyl group. And this would be a butyl group, a tetrabutyl group. This is isoleucine here. So these are all aliphatic residues. They have, they're hydrophobic. They don't have, they're they're not polarized groups, so they don't like to interact with water very well, okay? Then all the other and phenylalanine has an aromatic group, which is also relatively hydrophobic, even though aromatic groups, if you remember your chemistry, they have the pi electrons above and below the plane of the aromatic ring, and those are partially negative, so they're not as hydrophobic as these aliphatic amino acids here. And then we have tyrosine and tryptophan that have these other aromatic rings. This is a phenol ring, so it has a hydroxyl group here, which is relatively hydrophilic. This tryptophan has this nitrogen with a hydrogen bound, so it has a sort of a partially hydrophilic character as well. And then we have all these other amino acids here. We have these neutral polar amino acids that have side chains that have groups that can make hydrogen bonds with water. And these hydrogen bonds, these groups are hydroxyl groups that are shown here in serine and threonine, a thiol group, which is an SH group in cysteine, and then we have these asparagine and glutamine. They have an amide group, so it has an NH2 group here and a carbonyl group here that can accept or donate hydrogen bonds interacting with water, okay? And then we have these charged amino acids, 2 acidic amino acids, aspartate and glutamate, and 3 basic amino acids, histidine, lysine, and arginine, that can be positively charged. And they have different pKa's, okay? So lysine and arginine have pKa's relatively high, around 10 or 12 in the case of arginine. Histidine has a pKa around 6 or 7, so depending on the environment, it can be found in a protonated positively charged state or in a unprotonated neutral state, okay? And why am I mentioning this? So as well as these 5 basic and acidic amino acids, the 2 more amino acids can be unprotonated. Tyrosine, this phenyl group here, this hydroxyl group here it has a pKa around 10, so it can be deprotonated under certain conditions. And cysteine has a pKa around 8, so it can be found in a protonated or in a deprotonated state with a negative charge, okay? So these acid-base reactions involving these side chains are often very, very important in chemical reactions. Like we saw in that video, you saw that the aconitase was donating and removing hydrogen atoms using serine here and using histidine groups, okay, as acid and base catalysts in the active site of the enzyme, okay? So we have 4 different, so when we talk about the structure of a protein, so proteins are polymers of these amino acids. And when we talk about the structure of the protein, we can talk about the structure in many different ways. One general way is to classify the structure by talking about the primary structure of the protein is just basically the sequence of the amino acids of the protein. Okay, so it's the covalent structure. It's how the amino acids are attached together by covalent bonds. Okay. So that's the sequence of the protein. Then the secondary structure is how local sequences, sort of consecutive amino acids in the sequence, fold up and adopt sort of regular structural patterns. And we'll talk about those in a second. And then the tertiary structure is how these secondary structures interact with each other to make an overall globular form of the protein. And then often, many proteins adopt what we call quaternary structures, where more than one subunit, 2 or more subunits, sometimes similar subunits or different subunits, come together to make a complex of that is the functional form of the protein. So some proteins are made up of not just one polypeptide chain, but more than one, okay? So this is the sequence of a protein. So we have the amino acids come together to make a string of amino acids, okay? And these this polypeptide chain, which we call it, we can write the sequence down starting from one end of the protein and going to the other end of the protein. So we by convention, we write the sequence starting at what we call the amino terminus, or the first amino acid here, okay, which is shown here, showing this NH2 group, which really should be an NH3 group, because at physiological pH, this amino group is protonated, so it should be NH3 plus, okay? And so we have the alpha carbon, and the side chain here, which is represented with this R1, okay? So that's defining which amino acid it is. And then we have a carbonyl group, this carbonyl group, which has been condensed during the process of translation with the amino group of the second amino acid, making what we call this peptide bond here, okay? Which is the connection between one amino acid residue and the next amino acid residue. So we have a series of peptide bonds connecting all of the amino acids, and then we get to this carboxy terminus, where the last amino acid, its carboxy group, its carboxylic acid group, is left, okay? And here it's shown as COOH, but it should be COO with a negative charge, because at physiological pH, this is this has been deprotonated because the pKa of this group is around 2 or 3, and the physiological pH is around 7. So this should be deprotonated under physiological conditions. Okay. So as I said, this primary sequence folds up into secondary structures, and then these secondary structures come together to form what we call this tertiary structure, which is sort of like this globular form here. And here, every atom that's not a hydrogen atom has been shown. Actually, even the hydrogen atoms are shown here. The hydrogen atoms are these small spheres, and the large white spheres are carbon atoms. Okay. So secondary, tertiary, and quaternary structures are maintained mainly through non-covalent interactions. Okay, non-covalent interactions, they're relatively weak. They're much weaker than covalent bonds. Okay, and these non-covalent interactions can be classified. The major ones are hydrogen bonds, van der Waals interactions, dipole-dipole interactions, and also formal charge-charge interactions, and what we call the hydrophobic effect. Okay, and there's also disulfide bonds, which are actually covalent bonds between cysteine side chains. So these cysteine residues here, this SH group, this sulfur with a hydrogen bound to it, it can react with another cysteine residue in the same protein or in a different protein and undergo a redox reaction where the hydrogen atoms are lost along with their associated electron, and the sulfur atom makes a covalent bond with the sulfur atom of the other cysteine. Okay, and those are covalent bonds. Okay, and these non-covalent interactions, they have much weaker energies than covalent bonds. So covalent bonds between carbons, nitrogens, and oxygens, or covalent sulfur bonds that I just mentioned, have energies between 50 and 100 kilocals per mole. Okay, whereas these non-covalent interactions that I mentioned, hydrogen bonds, ionic interactions, dipole interactions, van der Waals interactions, okay, have energies that are less than 10 kilocalories per mole. But there are many, many of them, and so they will actually determine the overall structure, the final structure of the protein, based on a balance of these interactions between protein groups, intramolecular or intermolecular interactions with between amino acids in the protein or between one protein and another, versus the same kinds of interactions that the protein will make with the solvent, with water. So there's a balance. The protein has each interaction. It can either interact with another group in the protein, or it can dissociate. That interaction can be broken, and the same groups will interact with solvent. Okay, so that's a big challenge even until now, is to actually do the accounting, the energetic accounting, when you have literally thousands of these interactions occurring, and different possibilities of each atom possibly interacting with all other atoms in the molecule, or with solvent molecules. Okay, so that's a big challenge. But when we determine the structure of proteins, when we look at the atomic details of the groups in proteins, we see that the interior of the proteins are very, very closely packed. So there's, this is a representation of a model of a protein, and this net, this sort of cloud here, is representing the electronic density around the atoms, okay? And you can see that this is an arginine side chain interacting with different other groups, and there's a highly complementary interaction between different parts of the molecule to make a very, very well-packed interior of the molecule that's maximizing interactions. In this case, we have ionic interactions between a positively charged and a negatively charged group here, for example. And we also have van der Waals interactions, which occur only when the atoms of two different parts of the molecule come together and approach their van der Waals distances, the sum of their van der Waals radii. And when that occurs, you can stabilize these very, very highly compact forms of the protein. The actual folding of the protein will, especially between hydrophobic groups, will allow the water molecules that were before interacting with these non-polar methylene groups here, for example, in this arginine, even though the arginine has this positively charged guanidinium group here that has 3 nitrogens and they can make hydrogen bonds with water, it has also 3 methylene groups here, or 4 methylene groups here, that do not interact very well with water. Okay, so when this arginine is part of the interior of a protein, or when the protein folds, the surface of these methylene groups here is now no longer in contact with water, and the water molecules that were interacting with it now are liberated and interacting with other water molecules in the solvent, and the entropy, in this case, the entropy actually increases. And so there's an entropic element to protein folding that liberates water molecules that would otherwise be interacting with nonpolar groups in a not so favorable interaction. Okay, this is just showing another kind of interaction in the interior of proteins. Often you see these positively charged guanidinium groups of arginines interacting with aromatic groups. Like I mentioned before, aromatic groups have these pi electron shells above and below the plane of the ring, and they're negatively charged, and they will interact favorably with positively charged groups. So you often see this kind of interaction occurring. Okay, so let's go back and look at how these amino acids in the protein are interacting, come together to make this what we call a peptide bond. So here is the connection between one amino acid here, which has it's alpha carbon and its side chain, and another amino acid here that has its alpha carbon and a side chain. And they came together forming this peptide bond, which is basically an amide bond, okay? Has a carbonyl group and a nitrogen, okay? The important thing to notice here is that this nitrogen here has a lone pair of electrons that's not shown, it's not shown here, but this lone pair of electrons can be delocalized. Instead of being bound directly to the nitrogen atom, it can be delocalized between the carbon and the nitrogen, forming this double bond here. And this pair of electrons in the pi bond in the pi bond here can be delocalized to just the oxygen atom. So you have this resonance form this peptide bond that makes this carbon-nitrogen bond is partially a double bond, and double bonds, we remember from chemistry, they do not rotate as easily as single bonds, and they can be just the rotation between the trans configuration, which is shown here, and the cis configuration, which would be just a rotation of this bond 180 degrees, is very, very slow. And so basically, these bonds are basically always in the trans configuration, except in some circumstances where they can be found in the cis configuration if the subsequent amino acid is a proline. You can ask me about why that's the case, or you can think about it, and then we can talk about it later if you want. But almost all the time, between two amino acids, this bond is in a trans configuration. So there's a carbon alpha here, and then the trans, and then a carbon alpha here, okay? And it's always around 180 degrees, plus or minus 15 degrees. Okay, so this basically makes a restriction of the configuration of the peptide bonds between amino acids. Okay, but so we can, when we think about a polypeptide chain, we can think about, so all of these six atoms here, they all lie in the same plane. Okay, so when we look at a polypeptide chain, we can think of it as a string of planes containing the 6 atoms in the peptide bond, and they're connected, these planes are connected by the alpha carbons, which is the central carbon atom of each amino acid residue, where the side chain is attached, okay? And these planes are connected by single bonds here, which are given the names phi and psi. So the angles, the torsion angles of these two bonds here, the NH bond is called a phi angle, and the CC bond is called psi. And so basically, you can describe the configuration in three-dimensional space of a polypeptide chain if you know all of the phi and psi angles of each residue, okay? Theoretically. So you can basically imagine that some combinations of phi and psi are permitted, okay? But there will be some combinations that will bring into juxtaposition some groups, the van der Waals volumes of certain elements in contact with the van der Waals volumes of other elements, and you'll have what we call steric impediments, okay, and repulsions. So there are some combinations of phi and psi that are not permitted, and there are some combinations that are permitted in a sort of general way. And this can all be calculated theoretically, just taking into account the van der Waals radii of certain elements in certain chemical groups, and these van der Waals distances between these atoms. Okay, so when you do this calculation, this was done, I think, in the 1960s, 1970s. Okay, you get what we call a Ramachandran plot. Okay, which is basically a plot that looks at all the possible combinations between the angle phi, which is minus 80 to 180, and the angle psi. And basically, we can see that there are some regions, these regions here, these two major regions, plus this small region here, are the allowed combinations. And all the other regions are not allowed for almost all the amino acids, except for glycine. Glycine is the smallest amino acid whose side chain is just a hydrogen atom, and it's basically allowed almost everywhere, okay? But for most all of the other amino acids, these are the allowed regions. So when you look at a real protein, and you look at the structure of a well-defined structure of a protein, and if you plot all of the 5-psi combinations in that protein, you'll see that it basically follows this pattern. There are some that are outside of it, but these are all glycine residues. Okay, so what are these two regions? These two regions were recognized in the 1940s, I think, by Linus Pauling, making paper models of peptide bonds of small stretches of polypeptides and seeing how these things could fold up in a favorable way. And he basically found that this region here, if you have several amino acids, consecutive amino acids adopting this kind of conformation, it will form what he called an alpha helix. Until today, it's still called an alpha helix, which is a right-handed helix which is stabilized by hydrogen bonds between the carbonyl group of an amino acid at a position I or X and the NH group of an amino acid, 4 amino acids later on, so I plus 4. So this is the I plus 4 amino acid, and its nitrogen with its hydrogen is pointing down, making a hydrogen bond with this carbonyl group, which is pointing up. So in this alpha, you can see all of the amino acids, all of the carbonyl groups are pointing towards the C terminus of the alpha helix, and all the NH groups are pointing down towards the amino terminus of the alpha helix. And all of the side chains that are shown in purple here, they're all pointing out, okay, out on the perimeter of the helix. So as you go up the spiral, the side chains are all pointing up. So theoretically, this structure can accommodate almost any sequence because the side chains are not interfering with each other. Now, there are some amino acids that don't like to be in alpha helices for certain reasons. You can think about which ones. There are some amino acids that are found more at the amino terminus, at the very beginning of an amino acid, and there are some that are found more commonly at the end of an alpha helix, and some are found at the end of an alpha helix, etc., based on the specific characteristics of those amino acids. But theoretically, this structure can accommodate any sequence. Okay, and the NH groups and the carbonyl groups that want to make hydrogen bonds can make a hydrogen bond in the absence of water. So this structure can be found in an environment that's an anhydrous environment. So it can be found in the interior of a protein where there's no water, or it can be found in a stretch going through a biological membrane which is made up of phospholipids, which is hydrophobic as well. Okay, so alpha helices. So this, as well as being stable, as well as being permitted, okay, it also satisfies the hydrogen bonding requirements of these main chain groups, the carbonyl group and the NH group of the peptide bonds. Okay? Now, what's the other region here, which Pauling called the beta region, okay, which is a more extended region because the angles here are near 180 degrees or minus 180 degrees. And this basically has the polypeptide chain in a more extended region configuration where the NH groups are pointing up, pointing up and down, okay, every two every alternative amino acid, this NH group is pointing down, and then it's pointing up, and then it's pointing down, and then it's pointing up, and the carbonyl group is pointing up, and then down, and then up, and then down, as you go along the chain. And one, what he called a beta strand, okay, a beta strand can interact with another beta strand here in an anti-parallel configuration where the NH group of one strand makes a hydrogen bond with the carbonyl group of another strand, etc. Okay? So these two strands can come together in this anti-parallel manner to satisfy the hydrogen bonding requirements. And I'm not sure if it's very obvious here, but you can see the R's here. The R here is it's a large R, and here it's a small r upside down. Because basically these two strands form a plane. If you think of my fingers as the strands, and you're looking at it like this, two strands, okay? So you have the side chains alternatively point above the plane and below the plane, and above the plane and below the plane. So if you think of it as a sheet of paper, you could have several strands attached to each other, and all the side chains are above and below the plane, and the strands are connected in the plane by these hydrogen bonds, okay? And so this also, since the side chains are pointing above and below the plane of the beta sheet, because these strands come together to what is called to make beta sheets, it also can accommodate, theoretically, any sequence, okay? Because the side chains don't necessarily interfere with each other in the interior of the protein. And these strands can also come together in a parallel manner, okay? So they don't have to be anti-parallel, they can be parallel. So you can have what you call parallel sheets or anti-parallel sheets. You can have mixed sheets, okay? So now let's look at atomic models of, so this, I said dinucleotide. This is wrong. This is dipeptide. It should be called dipeptide. Dinucleotide is 2 nucleic acids. But okay. So anyway, so this is just like a model of an alanine dipeptide showing only the carbon atoms in green, the nitrogen atoms in blue, and the oxygen atoms in yellow, in red. Okay. And then if you look at an alpha helix, you can see with maybe, I'm not sure how many amino acids we have here, maybe 10 amino acids. You can see this helical form. You can see this is the backbone occurring here, and the side. You can see the side chains. And this is the side view. But if you look at it in an axial view, you can see that the side chains are all pointing outside, out of the on the perimeter of the alpha helix. Okay. And the inside of the alpha helix is basically, if you showed a space-filling model of this, there wouldn't be very much space here. Okay? So this is just the backbone of this alpha helix here. Okay? And this is what we call a ribbon representation of a helix. Okay? So often to simplify the model, because looking at this, you might say, well, what's really going on here? Okay? So we can simplify this sometimes when we just want to understand the basic path of the main chain going in three-dimensional space. We can simplify this with what we call a ribbon representation or a cartoon representation. Okay, so here we have like a little piece of the we have a helix here. It's a right-handed helix, and then it stops being a helix here, and there's a little extension. Okay. So here's what a beta hairpin, which is just basically 2 beta strands coming together to make a small beta sheet, and these 2 beta strands are connected by hydrogen bonds, as I showed before. Okay, and here's with a side chain. This beta hairpin, okay? This is just showing the backbone only, so we're not seeing the side chains anymore. And this is the backbone with hydrogen atoms. So here, the hydrogen atoms are shown in white, and you can see that they are actually pointing in the right directions to make these hydrogen bonds between the carbonyl group, the oxygen here, and the NH group here, okay? And we can have like what they call a stick model, which is here, a space-filling model here, okay, where every atom is shown as a sphere defined by its van der Waals radius. And then we can also show what's what they call a solvent-exposed surface. So basically, how much of the surface, of this surface, is accessible to water molecules. And you can calculate this imagining a water molecule being a sphere with a radius of 1.4 angstroms, and then sort of like rolling that sphere over the whole surface of the protein and seeing how much of the protein can be in contact with that sphere, okay? And so this is sort of like, that's what they call a solvent-exposed surface, okay? So when we look at proteins, at many, many protein structures, we can see that there's often, we see patterns that repeat themselves over and over again in different contexts. Okay, so like I showed before, we often have two beta strands in an anti-parallel manner associated, and they're connected by a very, very short, what we call a hairpin here, okay, a beta hairpin. So this would be two anti-parallel beta strands. But you could also have two parallel beta strands that are just basically, you have a beta strand here, then some kind of loop here. This loop could be just a straight loop, or it could have an alpha helix here, for example, or some other kind of structure here, and then another beta strand here, so they come in a parallel, they associate in a parallel manner, okay? So here we have another what we call a motif, a super secondary structure motif, okay, where a beta strand, an alpha helix, and then a beta strand. So that's what we call a beta alpha beta loop. And you can have many beta alpha beta loops coming together to make a large structure like this that basically has a beta sheet on the inside, okay? A parallel beta sheet on the inside, and on the outside a whole bunch of parallel alpha helices interacting with each other, okay? And this beta alpha beta loops can fold, if you imagine extending this, coming around, okay, this could fold up on itself and make a complete ring on the inside, which is basically shown here, where we have what we call an alpha beta barrel, where we have a beta sheet on the inside making basically a barrel on the inside, and on the outside a whole bunch of alpha helices, okay? There's another motif that's often encountered in calcium-binding proteins, which is called an EF hand, where you have these perpendicular alpha helices connected by a loop of 12 amino acids that have, at specific positions, they have negatively charged or electronegative groups that coordinate a calcium or magnesium atom in the center. Okay, and so many calcium or magnesium binding proteins, but surely not all of them, have what we call these EF hands. Okay, often we'll have two alpha helices can come together to interact together in what we call a coiled coil, where an alpha helix, if you look at the repeat, an alpha helix every 3.5 amino acids, 3.4 amino acids. It's more or less 3.5 amino acids. You make one rotation in the helix. So if this is a position A, B, C, D, and then E here, so it's 1, 2, 3.5, okay, making one rotation in the helix. So every 2 rotations in the helix is 7 amino acids. So 1, 2, 3, 4, 5, 6, 7, and then 1, 2, 3, 4, 5, 6, 7. So if every So if we name these positions A, B, C, D, E, F, G, the A position and the D position are always on the same side of the alpha helix. So if these positions are hydrophobic amino acids, then two of these helices will tend to interact with each other with their hydrophobic sides interacting to make what we call a coiled coil, okay? And then we have other kinds of topologies that are encountered. These are just some examples. And they have sometimes very interesting names that have been given. Up and down barrel, jelly roll fold. This looks like Brazilians will recognize it as a rocambole, okay? Some of the names come from the proteins in which they were first described. So this is a triose phosphate isomerase, or TIM barrel fold, that folds up like this, okay? So these names, sometimes you will come across them and you might scratch your head trying to understand what they mean. It's just a sort of a short form way of biochemists to give names to certain structures that have been observed many, many times. Okay, so when you say Tim Barrel, a biochemist or a structural biologist at least, he'll sort of have an idea of what you're talking about because he knows this kind of structure here. Okay, immunoglobulin fold. So this kind of structure is found in the domains of immunoglobulins, etc. Okay, dinucleotide binding fold or Rossmann fold, which is found in proteins that interact with dinucleotides, okay? So we can represent proteins with these ribbon or cartoon diagrams, which basically emphasizes the fold, the topology of the protein in three-dimensional space, okay? Which is much easier to interpret than looking at it like this, which is showing all the atoms, okay? But sometimes you have to, you want to look at this and you want to like zoom in onto the active site or a specific part of the protein. You have to represent it in this way. And this is a space-filling model, and this is the solvent-exposed structure. So we've gone from primary structure to secondary structure to tertiary structure. Just let's quickly talk about quaternary structure of biological units, where often two or more polypeptide chains will come together to form what we call a quaternary structure, or a protein complex. Okay, it describes the manner in which the polypeptide chains associate to form multimeric complexes, and they can often show some kind of symmetry. And like I said, they can be heteromultimers, where you have more than one different sequence of polypeptides, or what we'll call each polypeptide in one of these complexes. They're called subunits. Okay, so you can have heteromultimers or homomultimers. Okay, and these multimers, these quaternary structures, often exhibit structural symmetry, and so you can have what we call cyclic symmetry, where n identical subunits are associated by positions with a Cn axis of rotation, which is just a fancy way of saying that if you get this subunit here, if you rotate it by 180 degrees, you find the position of this subunit here. So this is symmetric, and there's an axis, there's what they call a C2 axis of rotation coming out of the page here in the center of this. And this is the C3 symmetry, C5 symmetry, C7, C9, etc. You can find all the different kinds of symmetry you can imagine in proteins. You can also have what we call dihedral symmetry, where 2 times n identical subunits are related by one Cn axis and n C2 axes. So basically here, it's sort of like, here we have insulin. It has D3 symmetry. So there are 6 subunits here, okay? They're colored in 6 different colors, 2 different shades of blue, 2 different shades of green, I'm not sure what this is, yellow or something, and 2 different shades of green or cyan, okay? And so there's one C3 axis, which is coming out of the page, and then there's 3 C2 axes. And the C2 axes, you can imagine them coming, one of them is here, another is here, and another is here. Okay, and if you just flip them in the page, you can see this. This is what they call dihedral symmetry. So this kind of dihedral symmetry is also very common in proteins. And you can also see tetrahedral symmetry, octahedral symmetry, icosahedral symmetry, etc. Okay, so that's where I wanted to end this. But if I have time, I could go on to talk about structure determination methods. But it looks like I've probably already used up an hour, right? Okay, so let's take a break now. And then I'll talk about structure determination methods in the first part of my second talk. Okay? Yep. Are there any questions? Has everybody seen this before? I imagine that a lot of you have already seen this before, like in your biochemistry classes. But some people have never seen this before. Okay? Yeah. So it was for you guys mostly, I think. Okay? Yeah. We have time to take a picture. It seems there's no questions. Everyone understood everything. Okay. Yeah. Okay. Yeah. I think we're going to take a picture now. Are we going to take the picture here, Fernando? Yes. Here? Okay. Go in the seats. Okay.